Embryogeny is a post-fertilization event in which the embryo is develops, which develops from the zygote, and also at the same time the entire ovule it transforms into the seed. So the entire step of formation of that seed is what will be considered as the process of like post-fertilization event, out of which only the development of the zygote is what is known as the embryogeny. So talking about embryogeny, it varies in the case of dicot and that of the monocot type of plant. So let's learn about the dicot first. Now, whenever we find that the first step is always going to be the same thing, that where the two the sperm nucleus that are entering the ovule, one will go and fertilize the polar nuclei, whereas the other one fertilize the egg forming the zygote. The polar nuclei being after fertilization, it actually transforms into the endosperm. Uh, that will be an entire different topic. Let's say we'll discuss that in with a different topic. Now today we'll be focused on only this formation of the zygote, and after that whatever development it takes place within. So as mentioned, in the case of dicot, we know that the plant is going to have two cotyledons. The seeds are going to have two cotyledons. So what we are going to see is the development taking place in this particular manner. First of all, the zygote that we find, it's going to transform into these two cells. The two cells are going to be unequal, means it's dividing mitotically, but these two cells are going to be unequal. One will be slightly smaller than the other. So if we talk about the direction of where which one is present, then the basal cell is going to be towards the chalazal side, and the terminal cell, or sometimes it is called the apical cell, is going to be towards the micropylar side. Means it is like wait a second, sorry, I mean I said it wrong. The apical cell is supposed to be in the chalazal side, and the basal cell is going to be towards the micropyle. Okay, now what we are going to find here is like these two cells are going to now undergo division. Okay, so when we are talking about division, so what we are going to find here is that the basal cell will first of all divide mitotically in like longitudinal plane. Is just going dividing one after another. It's in like that, means actually the division will take place in transverse plane. Means in the transverse plane it will actually divide, but they grow towards longitudinal side, means they keep on growing. You can say in the direction of this arrow that is shown here. In the direction of the arrow is actually going. The arrow is not meaning that, but actually it grows in this particular direction. It keeps on growing that. But the main division is that the two cells that are going to form from that, the basal cell is going to divide into these two cells first of all, out of which this particular cell will be considered as the main anchor, along with the rest of them acting as a suspensor. What is that? I'll say about that later. Now the apical cell on the other hand, it is actually growing into like 2 cell, then 4 cell, then 8 cell, like that. It just keeps on dividing itself. Definitely all division taking place here is mitotic. Now after division, what we'll be seeing here is that these two cells that are forming there, so these two cells are like they are the one that is transforming slowly into the embryo. Okay, means it is not the rest of the thing, but actually it is this particular one which is growing and developing into the embryo. Okay, so this terminal cell is the one which will transform into the embryo. So first of all 2 cell, then 4 cell quadrant, then 8 cell octant, like that. So like that way it just keeps on dividing, and until like it keeps on taking the shape of the embryo later on. Whereas the suspensor will just grow longer, of which it will form this 8 cell structure. When it is forming into this 8 cell structure, there is something will happen here, is that this particular cell, this one, the one next to this larger cell, because the larger cell is acting as an anchor, because this is the one which will go and like in a way it will borrow itself towards like we can say the part of the plant only, and will be considered as a hostoria or a hosturial type of cell. Now, if you're not familiar with it, then hosturial cells are actually capable of like stealing nutrients. It's like kind of like a parasite. It actually steals nutrient from the main body of the plant. So this is the one which is going to steal the nutrient, though it is not stealing nutrient, but rather for the development of the rest, it actually needs the nutrient to be absorbed. So this is going to borrow itself like a root. It will borrow itself towards the like body of the of that particular fruit wherever it is growing. Now it is necessary, otherwise the seeds will remain completely loose, isn't it? And until the seed is properly matured enough, it is not at all accepted to remain loose running like that. So it actually keeps itself attached and borrowed with the main fruit body with the help of this particular hosturial cell. But right after the hosturial cell, this particular cell that is there, this is known as the hypophysis. And this hypophysis cell, it will divide and will become the root cap and the shoot cap for the entire embryo. Because if you know or if you remember that when the root is forming or when the shoot is forming, it does have a protective capping on top of it, comparatively like sturdy kind of cells or tissues which actually protect itself, protect it from like getting any physical damage or anything. So those two cells, the two cap region, is actually formed from this particular hypophysis cell. Now, as the rest of the cells are dividing, so we are going to find that this suspensor, after getting at least that much long, like 8 cell structure, and so it will stop its division, it will grow no longer. Sometimes it can grow to 10 cell structure also, it doesn't like matter to that that much. Like once it is grown like that much big enough, then we are going to find that it will remain stagnant as that, and but the other side, the embryo, it will actually keep on dividing. Now let me tell you one thing. Up until this stage, means this formation of 4 cell embryo structure, or at least this much amount of formation, is what is known as the stage of pro-embryo. This is the pro-embryo stage. After that, the main embryo development, the rest of the embryonic development will take place. So for the rest of the development, what we are going to find is that the embryo is like dividing, is continuously dividing, and it will transform into this globular embryo shape. This like round shaped structure that we are seeing here. So this globular embryo is actually going to develop. You can see the other things are not changing itself, but this one, it just keeps on getting bigger and bigger and bigger, and it is that it will comparatively become very large. Now that globular cell will again now divide, but the division will be like more towards the edge, but that toward not towards the center. That's why it is going to form this particular type of structure. This is called the heart stage or heart shaped embryo, because it does represent the shape of that of the heart. And this is necessary because these two edges are actually what is going to like grow even longer and will transform into the cotyledons, the two cotyledons of the dicot seed. What about the embryo? The embryo is actually going to form at this particular location. The cells of this particular location is going to grow into that main embryo portion that we are familiar with. So right there you can see that portion is developing into the shoot meristem. This entire thing definitely is the embryo, but it is that particular portion which is becoming the shoot meristem. And this portion, this base portion, the one close to the hypophysis, is one which will transform into the root portion, means the root meristem, we can say, or it can be found right there. And then you can see the hypophysis is right next to it, so it is going to become the root cap, isn't it? So this is the one, the hypophysis is mainly the one which is transforming into the root cap, whereas the like the shoot meristem is growing on the other side, and the two cotyledons are completely covering it full. So this stage is what is known as the mature embryo stage. Here it is written as the torpedo stage also. So this is the stage we find. This is how we find that the dicot embryo, they grow. Let me show you a picture for that. So this is a stained sample you can see of a developing embryo, and you can see that the embryo is developing right there, and it is being connected with the main body of the plant right there. You can see the hostilial cell, you can see it right here, isn't it? Let me enlarge the image for you. So right there you can see this is the hostoria, and then you can see this is the like the rest of the suspensor. The hypophysis can be cleanly seen here. It's a globular embryo at this stage. You can see the heart shaped embryo at this particular stage, and thus then you can see the formation of the total the cotyledon, the two cotyledons stage you can see. And here the root is also developing, or the root cap. The suspensor is still there, you can see. So I mean the hostilial cell, it is completely borrowing itself to the growing seeds, right? And then finally this is a fully matured embryo where we can see the two distinct cotyledons can be easily observed. All the cells have got matured enough, and we can see this structure now like properly formed, developed. Now let's come to the monocot, embryogeny in the monocot. Now monocot, the first few stages are all same, okay, but then again the changes starts coming from like a little later. Means the initial development of the basal apical cells are all same, but then what we'll see that the suspensor means the globular embryo formation that will take place up until that point to some extent is same, but the suspensor is going to be not that long. It is going to be shorter. Means it is not dividing up to a 8 cell structure, but rather much shorter than that. Only a few cells only will see the full suspensor formation is going to take place. And terminal cell again is going to divide into the cotyledon. Oh yes, one thing I remember that this division initially will be of the terminal region. Means the basal will actually keep on remaining as it is bigger, and the other small cell is actually going to grow towards this side. Means the previous case it was growing out away from the basal, I mean the apical cell, but in this case it is going to go slightly towards the basal apical cell. Now what we are going to find here is that the suspensor is growing there, only a few cell structure, and then we find that the globular embryo is actually dividing itself like this way. So a globular shape embryo, near it is not fully globular, but near globular type shape embryo is actually developing right there. Now when it is developing into that particular form, it is going to grow slightly differently, because the globular embryo formation, once it takes place, it actually transforms into the heart shape embryo in the case of dicot. But here it will just become a bit larger and larger, because you can see like in this case the suspensor is actually near microscopic as shown here, isn't it? It is not a regression of the suspensor, but rather the rest of the things actually getting larger and larger. Now when the rest of the thing is getting larger, at that time from one particular side is actually the plumule or the shoot meristems are going to develop right there, and the exact opposite side is going to behave as the radical. The rest of the portion that is growing out is going to be the cotyledon. So how it's developing, see this cell that we are seeing here in this case, this particular side is developing into the plumule. If it is so, then this particular side is going to transform into the radical. Always the radical is going to grow next to the hypophysis, okay. Means here also we're going to find the hypophysis there, because the hypophysis has to transform into the root cap. So it actually going to grow next to it, close to it. So we'll find radical growing there, plumule growing here, and then radical will transform into the root, and the plumule is going to transform into the shoot, and the rest of the portion, this entire rest of the portion is going to develop into the cotyledon. So there you go, this is the structure of the embryo of the monocot, and in that case you can see that the the root, the radical here, the radical is right there, the root meristems you can say, and then the root cap region is right there. The cotyledons is around this area, the shoot is actually right within it, we can say. It's actually layered structure, so we can say it is like that, but the cotyledon and shoot is definitely two different structure. So the shoot can be found right there, and then it also has this scutellum, this particular structure called the scutellum. Now is like it's a diagnostic, like it's in a way this formation of the scutellum is actually an indication that this is a monocot type of seed. We definitely we can say that the formation of the cotyledon is one of the feature, but this can be seen in like a very large species, means the grass and all the species of plant that we know, they all have this scutellum formation. They are like, it's quite distinct in them. So this is like a shield for the entire growing embryo. So this is definitely one of the like very prominent feature that will be seen here in this case. Let me show you a sample, like a slide of like how it looks like in actually the seed. So there you go, that is actually the structure. You can see the cotyledon growing right there along with the shoot at that particular point. This is the scutellum. Let me show you here. Yeah, this is the entire scutellum, and there you can see this is the root growing right there. So this is the structure and development of the embryo in case of the plants.